Good afternoon, everyone. I'm Shi Yan. It's an honor to be here to share with you guys some of my findings in the final year projects. Today's presentation topic is a comparative study into the genetic relationship of two types of hepatocellular carcinoma using bioinformatics analysis. So to begin with, I'm going to introduce you some background information, and then the objective of the study, results of the study, and then based on those information, I will suggest the future directions and then the limitations. So let's get started. The hepatitis B virus is famous. There's 300 million people infected by this virus in the 2012. 30% of them are Chinese. It's important to notice that 7% of the total Chinese population is infected by this virus. That means every 14.2 people is a carrier of this disease. And the treatment is limited to the interferons, and this treatment is not able to clear the HBV viruses in the human hepatocyte, which lead to further. And the hepatocellular carcinoma is a major type of liver cancer. It is 0.8 million new cases in the year of 2012. 50% of them are Chinese. Survival rate of this disease is very low. The treatment is limited to one target therapy, only one drug FDA approved, and the surgical removal. Among those different type of HCCs, there's one type, this HPV-induced type of HCC is the highest occurrence and highest mortality. Look at those numbers, it's pretty scary. So in order to treat this disease or diagnose this disease, identifying those genes related to this disease are important. There are two ways, forward genetics and reverse genetics. Forward genetics is the way that we study the phenotype formation to identify a group of genes that are contributing to this disease. And the reverse genetics is the other direction that we try to see dysregulation or misregulation of a known gene. How does it contribute to the disease formation, and how much does it contribute? So among those forward genetic screening, there is one type is Sleeping Beauty. It's a beautiful name, huh? This Sleeping Beauty transposon system contains a transposon, is here in the red arrow here, and the transposons will recognize this specific sequence and cut the gene of interest out and paste it back into the chromosomal DNA, wherever there is a TA dinucleotide. So there are 200 million TA dinucleotide in the genome, so that provides a large sample pool for the insertional events. So in order to screen for the cancer genes, we use this T2-onc. We can call it like a troublemaker. This is the structure of T2-onc. It contains a strong promoter here to drive the pro-oncogene overexpression and the polyadenylation sequence to drive the premature termination of the tumor suppressor genes. So after we use these two events to induce the cancer, we will sequence those tumors. And where there is an insertion of this T2-onc sequence, where the insertional frequency is higher than the prediction, we call those common insertion sites, CIS. And those genes are nearby those common insertion sites, are therefore suggested as a candidate cancer genes. So there are different studies using different models to investigate the cancer genes. And here I use the wild type and TP3 mutant mice to represent a spontaneous type of HCC formation. This one published in Nature Biotech. And the second one is the using the hepatitis B virus surface antigen expressing mice. These mice are used to represent the HPV-induced type of HCC. The data published in Nature Genetics. And these two studies generate huge amount of genetic data. In order to study those data, I have to use the bioinformatics tools. The Cancer Genome Atlas is an internet-based, large-scale clinical data storage. It contains DNA and RNA sequencing data. And the CBAR portal is a software offered by the TCGA Atlas. It can calculate the frequency of occurrence of certain type of mutations and also the expression level of a gene. So these two are the DNA analysis or the gene level analysis. And the final, this one is the IP analysis. I use it for the signaling pathway analysis. And it based on the published literature results of a gene and the disease relationship, and calculate the statistical event and suggest the possible contribution of disease of my input genes, and also offer the network of common gene sets. So the objective of my study here is to first find the common genes that occur in the two types of ACC, and then the common molecular pathway involved in the two types of hepatocellular carcinoma, and I'll suggest some future directions. So the results part, I will start with a mice genomic data. There are 87 genes that have been identified commonly occur in both types of HCC, spontaneous and HBV-induced. So if we plot those 87 genes according to their frequency of occurrence in the mice genomic data, the y-axis represents the spontaneous type of HCC model. The x-axis represents the HBV-induced type of HCC. So we can see in the top right-hand side corner, those genes are commonly occurred in both types of HCC, are therefore those most important candidate genes for us to investigate. And if we put those 87 mice genes, common in mice genes, we put it into the clinical data, we see whether they are significant in the human. So we can see the size of the front represents the frequency of occurrence here. There are 5 genes that are most frequently mutated according to the clinical data. And more interestingly, if we see their type of mutation, they are frequently mutated, but what are the types of their mutation? It is the amplification in the copy number change. And the expression level in their mRNA is agree with this amplification in the copy number. So these two properties suggest that these 5 genes are those reassemble the property of pro-oncogenes. And more, if we look at the correlation of these 5 genes, among those 5 genes, 2 pair of genes have a strong co-occurrence. And this suggests they could be related in the molecular level either by protein-protein interaction or the downregulator of each other. So I try to investigate their correlations, but there is no known publication describe their correlations. So I use this computative software that suggests their possible correlations. Notice there are some straight lines connecting those genes. So there could be real connection in the protein-protein interaction level. And there are some genes that are connected to the known pathway, protein ubiquitination pathway, MAP pathways. So this provides a window for us to investigate in the future. So with that, I finished the gene and gene analysis of the 2 study data. Now I will try to show you some of the results in the signaling pathway results. This is the IPA analysis. As suggested by this software, hypoxia signaling pathway sent out during the analysis. And look at the p-value here is very significant. So that suggests the hypoxia signaling pathway could be the common pathway involved in the 2 types of hepatocellular carcinoma formation. They could be merged into one, possibly that results in the final tumor genesis. So that's my results, and now I will suggest some future directions. As always, I will start with the mice model data. So as you may recall, in the Dpyd and Zbtb20 are those 2 genes are frequently mutated in both types of mice models. So I think they are possibly the early driver genes of the tumor genesis event. What are the early driver genes? Early driver genes are those genes that are the regulator of DNA repair. Those genes mutation does not contribute to the tumorosis properties, but their mutation will promote the future late-stage gene mutations that actually contribute to the mobility inhibition property of the tumor. And what are the properties of early driver genes? The properties of early driver genes, since they occur in the early time frame, so they should have more time to develop into the early daughter cells. So these genes will have higher frequency when we sequence the tumor samples. And based on the same reasons, they should have higher frequency read count. So the Zbtb20 and Dpyd, as I previously described, they have this high frequency of occurrence. So they suggest they could be the strong candidate early driver genes for us to investigate. However, it is very interesting to notice that these 2 genes have their very important role in the mouse HCC, but when I try to look at their occurrence in the human HCC data, they are not such strong co-occurrence. So this event can be explained by two reasons. Possibly the sample size of my clinical data is not big enough for generating a unbiased clinical data, or there are some real differences between the mice hepatocyte and the human hepatocytes. So further research can investigate their differences in signaling pathway or use the reverse genetics to identify those genes. And let's see the human clinical data. Previously, I mentioned there are 5 genes that are very significant in human clinical data, and 4 genes are those normal genes. There's no literature describe the relationship between these 4 genes and the HCC formation. And about WWP1, there's only one publication describe its relationship in the HCC tumorigenesis event. So that provides a huge opportunity for us, the researchers, to investigate their function in HCC in the future. And now I will take some time to explain the limitations of the study. First, in the HBV-induced type of HCC model, I used the HBV surface antigen expressing mice to represent the HBV infection scenario. But actually, how does HBV contribute to the HCC formation? It's a complicated event, either by the direct integration of the viral genome into the hepatocyte, or it could be the expression of the HBV proteins that indirectly cause or promote the mutagenesis event in the hepatocyte. But in this case, I only use one, the HBV surface antigen expressing, so that's limit the results of my analysis. And also for the spontaneous type of HCC, I use here is majority of the mice sample I use is the TP53 mutant mice. So that could contribute a bias towards the TP53 and DM2 pathways. So those genes are actually may be not actually spontaneous. They have some bias. And the third limitation here is the sequencing of those tumor samples. Both of the study only use DNA sequencing. So they identify those T2-onc, remember those troublemaker sequence. So they use the sequence of those troublemaker insertion sites to suggest those candidate cancer genes. But as we know, the dysregulation in the DNA level does not necessarily mean they will be following mRNA dysregulation or mutant protein production. So I think in the future, RNA sequence, at least RNA transcript sequencing is enough, necessary for us to have a confident suggestion for the candidate cancer gene. And I think, yeah, that's all about my presentation. Before I end, I'd like to offer my special thanks to Supervisor Dr. Vincent King for his patient guidance and enthusiastic encouragement for me. And also the lab mates and some people from the other group provide the software support and also some knowledge support. Thank you, and thank you for listening. Thank you.
Can I go back to your limitation page?
Yeah, you can use this.
So you mentioned about the So you mentioned about the inadequate insertion mutagenesis analysis that the DNA sequencing will not be able to reveal the changes. So, to my knowledge, this type of insertional mutagenesis always reduces the effect in terms of the knockout. Am I right?
Yes, that is kind of right.
And why do you think DNA sequences is not enough to reveal the function of this insertion?
So maybe I need to go back to here. This T2-onc does not only make the knockout. It also make a upregulation of the pro-oncogenes. There's two functional screens, loss of function and gain of function. And this is pretty new. In this beginning of this month, November, there is one publication. Actually, they sequenced a small group of the tumor, and 80% of those samples does not in the RNA sample does not have the dysregulation, but those previous samples they suggest to be candidate cancer genes, they have those insertion in the DNA level. So in the RNA sequencing, they see actually 80% of the transcript does not have the significant modifications. So I guess,
what is the reason?
yeah, there's investigation in that. So I guess maybe the cells, they have their regulations. So as you may know, this one, this sleeping beauty thing is from a fish, I think. So their basic kind of regulation. Human is a higher species, so they may have a more complicated regulation, either by exon splicing. So the insertion of this in the exon 1 may necessarily contribute to the following, the transcription translation event. So DNA level is not necessary. We have to see whether RNA level changes and also the protein level changes.
I think I need to step in. When the T2 inserts itself into a gene suppressor, it's not a knockout. It's only given one allele. Okay? So you need to mutate the other allele, which sleeping beauty rarely does. So you need a de novo loss of LH. Okay? And then that becomes a true loss of innocence. Okay? So when he's talking about the 80% of the genes that will not match, it makes a lot of sense because we're only getting one allele in some of those cancers. So are they picking up the real suppressor genes, or are they picking up something else?
So what's the root cause for the tumorigenesis then?
Well, naturally, you
only
get one allele, so you rely on the other allele being mutated naturally through kind of mutation.
That event